Life on Earth is amazing. It's so complex, with tiny machines doing incredible things inside every cell. But how did it all start? That's a huge question, maybe the biggest one in science. The RNA World hypothesis is a major idea that tries to answer part of that question. It tries to explain how life might have kicked off, how the very first forms of life could have begun before everything got so complicated. Think about how living things work today. You have DNA, right? DNA is like the master blueprint, the instruction manual for everything. It holds all the genetic information. But DNA doesn't do anything itself, it just sits there storing information. To actually build things, to carry out tasks, to make things happen, you need proteins. The proteins are the workers, the enzymes that speed up chemical reactions, the structural components. They're like the tools and the builders. Here's the problem: DNA needs proteins to function. Proteins are needed to copy DNA, to repair DNA, to even read the DNA instructions to make more proteins. And proteins, in turn, need DNA. The instructions for making every protein are encoded in DNA. So you have this classic chicken and egg situation. Which came first? Did you have the information storage system DNA first, or the workhorse molecules proteins first? It seems like you can't have one without the other. This is a real puzzle for scientists trying to figure out how the first life forms began. This is where the RNA world hypothesis steps in. It suggests that there was a time, very early in Earth's history, when RNA ruled. RNA stands for ribonucleic acid. It's a molecule very similar to DNA, but with a few key differences. The idea is that RNA molecules could do both jobs. They could store genetic information just like DNA does. And crucially, they could also act like enzymes, speeding up chemical reactions just like proteins do. So, in this early RNA world, you wouldn't need separate DNA and proteins. RNA could do it all by itself. This solves the chicken and egg problem because RNA could be both the chicken and the egg. It could be the information in the worker. Let's break down why RNA is such a special molecule and why this idea makes so much sense. First, let's look at what RNA is. It's made up of building blocks called nucleotides, just like DNA. Each nucleotide has a sugar, a phosphate, and a nitrogenous base. In DNA, the sugar is deoxyribose. In RNA, it's ribose. That's one difference. Another difference is one of the bases. DNA uses A, T, C, G, adenine, thymine, cytosine, guanine. RNA uses A, U, C, G, adenine, uracil, cytosine, guanine. So RNA has uracil instead of thymine. Also, DNA is usually a double helix, like a twisted ladder. RNA is usually single-stranded, but it can fold back on itself in incredibly complex ways, making all sorts of three-dimensional shapes. These shapes are really important for its function, just like the shapes of proteins are important for their function. The really big deal about RNA, the game changer for the RNA world hypothesis, is its ability to act like an enzyme. For a long time, scientists thought only proteins could be enzymes. They were called catalysts, molecules that speed up specific chemical reactions without being used up themselves. But in the 1980s, scientists like Sidney Altman and Thomas Cech discovered something amazing. They found that certain RNA molecules could also act as catalysts. They called these special RNA enzymes ribozymes. This was a huge discovery, and they even won a Nobel Prize for it. The discovery of ribozymes was like finding the missing piece of the puzzle. It showed that RNA wasn't just a messenger or a helper molecule. It had the power to do work, to make things happen, just like proteins. This meant that an early Earth where life was based solely on RNA was not only possible, but suddenly seemed very plausible. Let's talk about some real-world examples of ribozymes that still exist today. The most famous one is probably the ribosome. What's a ribosome? It's the cellular machine that makes proteins. It reads the instructions from messenger RNA, mRNA, and links amino acids together to build a protein. For a long time, people thought the proteins within the ribosome were doing all the catalytic work. But it turns out that the core catalytic function, the part that actually forms the peptide bonds between amino acids, is performed by ribosomal RNA, rRNA. The ribosome is essentially a giant ribosome, with proteins mostly acting as a scaffold or helper. This is incredibly strong evidence for the RNA world because it means the most fundamental process of life protein synthesis is still RNA dependent at its core. It's like a relic from an ancient past, still active in every cell. Another example is RNase P. This is an enzyme involved in processing transfer RNA tRNA molecules, which are crucial for carrying amino acids to the ribosome during protein synthesis. In bacteria, RNase P is a ribozyme. The RNA part of it does the cutting. There are also self-splicing introns. These are bits of RNA that can cut themselves out of a larger RNA molecule and then stitch the remaining parts back together, all without the help of any proteins. This shows RNA's ability to manipulate other RNA molecules. So RNA can store information and RNA can do work. What else? Well, RNA also has the potential for self-replication. Imagine an RNA molecule that could not only catalyze reactions, but also catalyze its own copying. That would be the ultimate self-sufficient molecule. Scientists have actually made progress in the lab showing that RNA enzymes can copy other RNA molecules and even small parts of themselves. While we haven't yet created a truly robust self-replicating RNA molecule in a test tube, the principle has been demonstrated. This possibility means that in an early RNA world, RNA molecules could have made copies of themselves, and those copies could have evolved. Mistakes in copying would lead to variations, and some variations might be better at copying themselves or doing other useful tasks. This is the basic engine of evolution. Beyond ribozymes, there are other clues hidden in our cells that point to an RNA world. Look at the molecules that power life. ATP adenosine triphosphate is the main energy currency of the cell. NOD and FADH2 are also critical for energy transfer. What's interesting about all these molecules? They all have an RNA-like component, an adenine base linked to a ribose sugar. This isn't just a coincidence. It suggests that these fundamental energy molecules might have evolved at a time when RNA was central to life, maybe even serving as cofactors for early ribozymes. It's like finding old parts from a bygone era still doing important jobs in a modern machine. Even DNA synthesis itself gives us a hint. When DNA is copied, the enzyme that does the copying, DNA polymerase, can't just start from scratch. It needs a little helper. It needs a short piece of RNA, called an RNA primer, to kick things off. This RNA primer is later removed and replaced with DNA, but the fact that RNA is still required for the initiation of DNA replication suggests that RNA came first. It's like the training wheels for DNA. So if we put all this together, how might the RNA world have looked? Imagine the early Earth, billions of years ago. It was a very different place. No oxygen in the atmosphere, lots of volcanic activity, lightning, and intense UV radiation. In this primordial soup, a mix of simple chemicals in ponds or oceans, basic organic molecules started to form. Over time, these simple molecules, like amino acids and nucleotides, might have linked up to form longer chains. Maybe clays or mineral surfaces helped to concentrate these molecules and catalyze their formation. Eventually, some RNA molecules formed. These weren't perfect, long, precise RNA molecules like we see today. They were probably short, messy, and random, but by chance, some of these RNA molecules might have folded into shapes that allowed them to do something useful. Maybe one could help another RNA molecule link up, maybe another could cut a bond. And then, the magic step, maybe one could help make copies of itself, even imperfectly. Once that self-replication started, evolution could begin. RNA molecules that were better at copying themselves, or more stable, or better at catalyzing useful reactions, like making more of their own building blocks, would become more abundant. This is natural selection at work, even at this very basic molecular level. These early RNA populations would compete and evolve. Over millions of years, they would get more sophisticated. They might start working together, forming more complex networks of interacting ribozymes. At some point, proteins would have entered the picture. Perhaps some ribozymes started to make short chains of amino acids. At first, these small proteins might have just been helpers for the RNA, improving the efficiency of the ribozymes. But proteins have a much richer chemical diversity than RNA. They can fold into more intricate shapes and perform a wider range of catalytic tasks with greater speed and precision. So there would have been an evolutionary advantage to starting to use proteins for more and more of the cellular work. And then came DNA. Why switch to DNA? Well, RNA is a pretty good molecule, but it has some downsides. It's less stable than DNA. The ribose sugar in RNA makes it more reactive and prone to breakdown, especially in the presence of water. DNA, with its deoxyribose sugar and its double-stranded structure, is much more chemically stable and resistant to degradation. It's a much better molecule for long-term information storage. So over time, as life got more complex and needed a more reliable way to store its ever-growing genetic blueprint, DNA would have been selected. So the proposed transition would look something like this one. In RNA world, RNA molecules handle both genetic information and catalytic functions. RNA then takes on its modern roles as an intermediary mRNA, a transporter tRNA, and a core component of the ribosome rRNA. These proteins become the main workhorses of the cell, performing most of the catalytic and structural jobs. This whole process, from simple RNA to the complex DNA RNA protein system we see today, would have taken immense amounts of time, evolving step by tiny step. Of course, the RNA world hypothesis is still a hypothesis. It's a very strong one, backed by a lot of evidence, but there are still challenges and unanswered questions. Science is all about asking questions and seeking answers, and this hypothesis gives us a framework to do that. One of the big challenges is how the building blocks of RNA, the nucleotides, formed in the first place on early Earth. This is part of the broader question of abiogenesis, the origin of life from non-living matter. How did simple chemicals spontaneously form sugars, bases, and phosphates, and then link them together into nucleotides? And how do those nucleotides then link up into long RNA chains? We call this the prebiotic soup problem. While scientists have shown in lab experiments that some of these steps are possible under early Earth conditions, getting all the pieces to come together efficiently and in the right forms like only D-ribose, not a mix of D and L forms, is still a hurdle. Another challenge is the stability of RNA itself. As mentioned, RNA is less stable than DNA. In a hot, chaotic early Earth, how could these crucial RNA molecules have persisted long enough to evolve and replicate effectively? Perhaps they were protected inside primitive compartments or on mineral surfaces. Then there's the question of specificity. Early ribozymes might not have been as efficient or as specific as modern protein enzymes. How did they manage to perform the necessary reactions well enough to drive the emergence of life? This suggests that early life might have been very inefficient and slow by today's standards. The transition from an RNA world to a DNA protein world is also complex. How exactly did RNA learn to make proteins? How did the genetic code, which links specific RNA sequences to specific amino acids, get established? This code is universal in all life today, suggesting it evolved very early and was locked in. And how did DNA take over as the genetic material? These are big evolutionary jumps that are not fully understood. It probably wasn't a sudden switch, but a gradual process of coevolution. Some scientists even propose a pre-RNA world. They suggest that maybe RNA itself wasn't the very first self-replicating molecule. Perhaps there were even simpler genetic polymers like PNA peptide, nucleic acid, or TNA-threose nucleic acid that were more robust or easier to form under early Earth conditions. These ancestors to RNA might have then paved the way for RNA, which then led to DNA and proteins. It's like a deeper level of recursion trying to find an even simpler starting point. This just shows how complex the origin of life truly is, and how many possibilities scientists are exploring. Despite these challenges, the RNA world hypothesis remains the leading contender for how life might have emerged. It provides a coherent framework that solves the chicken and egg problem, explains the pervasive role of RNA in modern cells, and is supported by a growing body of experimental evidence. It's a powerful idea because it connects disparate observations into a single elegant story. The significance of the RNA world hypothesis extends beyond just understanding our planet's past. It influences how we think about life elsewhere in the universe. If life on Earth started with RNA, could a similar RNA-based system arise on other planets or moons? It guides astrobiologists in their search for extraterrestrial life, suggesting that the fundamental chemistry of self-replication and catalysis might be common ingredients for life wherever it arises. The research continues. Scientists are trying to synthesize RNA nucleotides more efficiently under simulated early Earth conditions. They are trying to evolve more complex and efficient ribozymes in the lab. They are trying to piece together the detailed steps of the transition from RNA to proteins and DNA. Every new discovery adds another piece to this incredible puzzle. In the end, the RNA world hypothesis tells a story of self-sufficiency. It says that at the very beginning, a single type of molecule had enough versatility to kickstart life. It could hold the instructions and it could carry out the work. From that humble dual-purpose beginning, the astounding complexity of life as we know it gradually unfolded. It's not a complete answer to everything about life's origins, but it's a profound and elegant step towards understanding our deepest roots. It reminds us that even the most complex systems can begin with surprisingly simple, yet incredibly clever, solutions.